We're here at the CROI in Boston, and we're here with John Coffin, who's from Tufts University, and he's a leader in the science, the basic science field, and often does the press conferences that involve those particular prominent presentations that are going to be given at the CROI. And you were chair of the CROI, so I mean, you're involved with this event, but more than that, you're involved with the science, and you find it exciting, and that's why you're here today, to tell us what basic science is and why should people be interested in and understand it better.
Well, thank you, Fred, and thank you for the opportunity to come and tell people about this. It is indeed a very exciting topic, even aside from its obvious and tremendous importance in curing people, or trying to cure people, or at least preventing people from dying from HIV infection. The understanding the nature of our enemy in this case is actually a very fascinating part of science, and studies on HIV have actually informed many other fields of science now in many important areas. It's understanding evolution, understanding immunology, understanding very basics of how genes work, and of understanding how to deal not only with HIV, but with many other virus infections. I think, for example, the very recent dramatic advances in HCV infection, which is now a curable disease, it really builds on the experience with developing HIV therapies, and those build on, in a very important way, understanding the basic science of HIV, and before that, of understanding the basic science of retroviruses in general. In fact, I grew up scientifically in the era before HIV. I was a graduate student in the lab of Howard Temin, where the reverse transcriptase was discovered, and of course, we had no idea at the time that there was even a human retrovirus at all, any retroviruses of that group infecting humans. We knew they were very important agents infecting chickens and mice and cats and a number of other species, and that there were important causes of cancer and certain kinds of other diseases. And because of that, there was a very intensive research effort, funded largely by the National Cancer Institute years ago, to understand how retroviruses work and how they cause cancer, because it was thought, and in fact it was thought correctly, that they could provide very important basic information for understanding how cancer is caused, even if it's not caused by a virus. And that turned out to be true. The research on retroviruses before the advent of HIV in 1983 was really the first research that revealed the fundamental mechanisms by which cancer is caused. And that's because retroviruses are in fact very special things in biology. They interact with their hosts in ways that no other virus or no other infectious agent does. They work by causing a copy of their genes, of their genetic information, to be inserted into the infected cell in the form of DNA, and to be inserted in such a way that it is there permanently until retroviruses are forever, actually. Used to be like they used to say about the difference between love and herpes, of course, and that herpes is forever. Well, the same thing is true of any retrovirus. Once it infects a cell, it's there forever, as long as that cell lives. And if that cell divides, the virus information divides right along with it. And that's actually one of the things we very recently discovered about HIV infection, our understanding of how HIV persists in the face of very effective therapy has been substantially advanced. There's going to be a lot at this meeting about that, actually. That we now know that HIV-infected cells in a body that is, in a person that's being treated with effective therapy, those cells, although the therapy is very effective against preventing the virus from infecting a cell, once the virus infects the cell, once it gets past that, then these drugs are completely ineffective. We have no drugs that can really target the virus information when it's present in the cell that's already been infected. So if one individual that goes on therapy has tens of millions or hundreds of millions of infected cells in him at that time, those cells die, but not all of them die. And a certain fraction of them survive. They carry the virus genetic information. A small fraction of those carry the virus genetic information in a way that it can be reappear even years later. And we've known that for quite a long time. What we've recently learned is not only do those cells sit there with the genetic information, but they can actually divide into small populations, which we call this clonal expansion. A clone is a collection of cells that have derived from the same original cell by division. So, we have recently discovered that these cells can divide substantially, and they can not only make it, unfortunately, that makes it all the much harder to try to get rid of those cells, even if we had a way to do it, because the numbers now we know are much larger than what we thought before, and they're capable of self-propagation, so that even if you could treat an individual to get rid of 99% of those cells, the ones that were left, like analogous to a cancer, actually could then grow back again. And
So all this information is so collective. It's like some people are always looking for the magic bullet, or they're looking for this to simplify this thing.
Right.
It is a very complicated.
It's a very complicated problem.
But we don't. The person out there in our audience doesn't need to know all the complexities.
Right.
Just kind of a similar. We use a lot of pictures, like cartoons, so to speak, to kind of describe what the virus is doing and so forth. So what we've learned over the last number of years has been massive.
Yes.
In so many ways, like you said, it informed all the other various ends of science. But it's also headed towards we're really seriously looking for a cure now.
We're seriously doing that. We don't yet have any agreement on what the best way to do that right now is, but there are many avenues that are being pursued. The one that's most popular, although perhaps most difficult, is what's called a shock and kill strategy. The idea is that a cell, as I said, that has the viral genetic information in it is growing in the body, is usually not expressing that genetic information. As far as the virus is concerned, it's just sleeping. It's completely quiet.
We used to call them compartments, or hidden compartments, or something like that.
There's compartments, and there's also just individual cells that where if the virus genes were being expressed, if the cell was actually making virus all the time, the thinking is that cell would not live very long. It would not live very long, both because the virus itself can kill it, but also the immune response of the person will recognize that cell if it's making what are called antigens, viral proteins, virus particles. If the cell is able to make those, then the immune system of the individual would wipe those cells out. And the idea of these shock and kill strategies is to figure out a way to get all of the cells that have these sleeping, what's called latent or sleeping viruses in them, to actually wake up and start making virus, and then the immune system of the host could get rid of that. Or perhaps we could, at the same time, introduce some kind of a vaccination strategy to give the immune system a boost, because in people...
Or a combination thereof.
A combination, exactly, because people who've been treated for HIV infection for a long time, the immune response actually tends to go away because they're not seeing, it's not being stimulated anymore. There's not enough virus around to keep it active. So the immune system is designed to respond to an infection when the infection is present, but then to turn itself off, and it can recognize it, but then to turn itself off when the infection is gone. Right. And so that's what tends to happen in the case of HIV treatment. when the infection is suppressed by the combination therapies that are used today and suppressed very effectively. When it's expressed, the immune system turns itself down, and it needs to be woken up again in order to participate in the cure process if we're going to be able to do that.
So that's the shock piece of it.
That's it's the shock piece is getting the virus, is shocking the cells so they'll start making virus again, and then the kill piece is relies on an immune response that will actually recognize those cells and sweep them out like it's doing actually during untreated infection. Cells that are infected during untreated infection are being killed by the immune response. It's just the immune response isn't quite quick enough. The virus has figured out to do this fast enough that it can get some virus out of each cell before the immune response gets in and kills the cell.
Now, again, we talk about compartments, but they're not really like a pocket over here in your brain. But it could be. I mean,
there are some anatomical compartments. There are also
the brain and so forth,
cell type compartments that are cells that are distributed in different places, but
they're
all together that group of cells can be called a compartment as well.
It's not a little box or anything.
Right,
it's
right.
the type of cell.
Right, right. Well, it can be both.
Or both.
It can be both. It can be cells in the brain, it can be cells in lymph nodes, it can be cells in the spleen or whatever. But then there are also different types of cells that people talk about, different types of cells like T cells, for example, which are the cells that are
all over the body.
Cells are all over the body that are sort of the first sentinel, some of the first sentinels for infection. That's T cells that that can sniff out cells that are making virus and kill them, hopefully before they have a chance to make virus, but actually that, in the case of HIV, it's figured out how to get out just ahead of the cells that are coming around trying to kill the cell that's infected.
It's a very devious virus, but
devious.
But there's a lot of doctors. There's been a huge effort to take this basic science that you and so many of your colleagues have developed and kind of transform it into action, into the Delaney collaboratories and any number of cure collaboratories that are being out there right now. So it's exciting times, different than in the old days when there was this unaware situation of the way the virus was being devious that you've now explained.
Every year we learn more and more about how devious the virus can be, because the cells, our bodies throw up an enormous number of roadblocks. So this is a major thrust of research for quite a number of years now. Our bodies throw up an enormous number of roadblocks, specifically to try to prevent the virus from doing what it wants to do. And the virus, with every roadblock, the hosts for these viruses, like us or any other animals, have thrown up. The virus has figured out some very clever counter strategy to get around it. And this is called the host virus arms race. It's literally you develop a missile, and the virus develops an anti-missile, and you develop an anti-missile, and so on and so forth, and it just keeps going back and forth and back and forth. And we keep seeing every year at CROI, we see some new reports of new ones. There's a particularly exciting one of these that's being reported this year. So
we had the Mississippi baby, and everybody had hopes that was going to be something to learn a lot. Well, we did learn a lot.
We did learn.
And the same with the Boston patient, which I'll actually be interviewing with the doctor, Dr. Tim, who is
Tim Henrich,
Tim Henrich is a really great guy, and we always do an update with him because it's exciting. And I think the other piece of it is that we have to ask people to participate in trials. So we have to make this interesting. From people like yourself who really express how important it is. You know, a lot of the basic science is research in animals, lab animals, and so forth. But right from that, we take it into humans, and people have to be ready to do that.
And it's important to realize that not all trials are sort of directly cure trials. Many of them are just are directed to getting the important information we need in order to move one baby step toward that. And right now we're moving in baby steps. I think we're moving in that direction, but it's a
lot to learn.
There's a lot to learn. We really, and as I like to say, sometimes the best science isn't always the best news as far as as far as the ability to apply the information that we get to, but it does at least save us from going down dead ends, which is very important.
But you started your career in immune science.
Well, actually, I started my career in viruses. I was working
Viruses.
with Howard Temin, who's who won the Nobel Prize for the discovery of reverse transcriptase in retroviruses.
So what a wonderful start for your career.
It was a very good start. Yes.
I think, and like we talked earlier, I always like to interview the young investigators, and I always see
You've
this.
missed that opportunity with me by quite a few years, unfortunately.
We're doing it retrospectively.
There you go.
Yeah. But there's so many great people we've interviewed over the years, and I know a number of them were by leading doctors, and it seems the leading doctors always try to get somebody that's really, really out there and great science and so forth, and they get them a lab right away and so forth. But it's very competitive, and I think the one thing we have to know, and everyone needs to know, is how exciting and how challenging and how maybe rewarding it is down the line.
Right.
Because even though you learn a little piece, it may not be a Nobel science worthy,
Right.
but it could be something that would lead the science to eventually a cure, which is exciting in the promise of that.
It's important to understand, for example, in the case of the work that was done in the lab where I was a student, that knowing about reverse transcriptase and made it possible to discover HIV in the first place, because that was how it was identified, was because of this enzyme. And then it made it possible to treat HIV. We still use anti-reverse transcriptase drugs as part of the main line of treatment. And so these things just progress along, and it's very important to have that base to build on.
Right. Well, I think this has been very helpful. I think we've kind of covered a lot of territory in a very basic way and with basic science. But down the line, we want to make sure people are ready for the trials, ready for those that will lead us either to better antivirals or possibly a cure. And they may not be a direct reward to that patient. We inform them so. But it will be an informed research project that will answer some questions.
Yes,
to maybe a cure.
That's very important. We can't progress without the help of patient volunteers.
Yeah, right. Well, thank you so much. I appreciate it. Thank you, John.
Okay.
All right.
My pleasure.